Welcome to the deep dive. Today, we're tackling something really fundamental, how our brains actually learn, how they store information.
Yeah, the real nuts and bolts.
Exactly,
Yeah.
understanding the mechanics, which is absolutely vital if you're doing any kind of serious learning. Thinking particularly about, say, preparing for the UPSC exams.
Now we look.
We've been looking at some fascinating research highlighted over on Singularity Hub based on a new study in Science.
That's the one.
Hmm.
And it's quite a shift in thinking, actually.
What's the core idea?
Well, the main takeaway is that neurons, which we often think of as, simple computational units, are much more complex internally.
More complex how?
Different parts of a single neuron seem to follow totally different rules when it comes to learning, which really challenges the traditional models of how brain networks operate.
Okay, so today we'll unpack that famous phrase, fire together, wire together.
Right.
Dig into dendrites and synapses, look at this specific study, this discovery about localized learning rules within a neuron, and then talk about what it all means for memory, maybe even AI. Sound good?
Sounds great. Let's dive in.
Okay, so that phrase, fire together, wire together, that's been the standard for a while, hasn't it? Can you bring that down first?
Sure. It's the basic principle of Hebbian learning. Essentially, if two neurons are active at roughly the same time, the connection, the synapse between them, gets stronger.
So that's how associations form, how we learn.
That's the foundational idea, yeah. Repeated coactivation strengthens the pathway. But what this new research highlights is it's not just about the connection between neurons. The neuron itself, particularly its input branches, the dendrites, they're not just passive receivers.
Okay, wait, dendrites. Those are the branchy bits that collect signals.
Exactly. Think of them like the tree branches gathering information. The old view is kind of like they just funnel everything down to the cell body.
But the new view is...
The new view, supported by this study, is that these dendrites are incredibly active. They're performing computations themselves, like mini processors on each branch, analyzing inputs before they even reach the main neuron body.
Wow. So it's not just neuron-to-neuron connections changing. The processing within the neuron is changing too, and differently in different places.
Precisely. Different compartments, especially these dendritic branches, seem to have their own rules for adapting during learning.
That sounds incredibly complex to manage inside one tiny cell. How did they even figure this out? You mentioned a study from UC San Diego.
Yeah, published in Science, they basically watched synapses, the connections, in action, in real time, in a mouse brain.
Watched them? How?
They used some pretty advanced genetic tools and imaging techniques. It let them monitor the activity of individual synapses in a part of the brain-controlling movement.
Okay.
So they had these mice learning motor skill. I think it was pressing a lever to get a reward. Classic learning task.
Right.
And as the mice learned, the researchers saw that synapses on different parts of the very same neuron were changing in very different ways.
Not all changing together.
No. Some synapses seem to be focused on syncing up really tightly with their immediate neighbors, forming these local clusters. Others seem to adjust based on the neuron's overall activity level. Different rules for different locations.
So even within one cell, learning isn't uniform.
Exactly.
Okay.
One of the study authors, William Jakewright, he put it nicely. He said, "Our research provides a clearer understanding of how synapses are being modified during learning. It just shows the nuance,
Yeah.
It's not a simple on-off strengthening everywhere."
And you mentioned two types of dendrites involved, apical and basal. Can you paint a
Right.
picture of those?
Yeah, okay. Imagine the neuron is like a tree. The apical dendrites are like the higher, sort of sparser branches reaching up towards the surface of the brain cortex.
Okay, higher branches.
Right. And the study found that the synapses on these branches, the apical ones, followed a rule of rapidly syncing up with their direct local neighbors, like creating really tight local networks right there on that branch.
Very localized learning.
Very localized.
Mm.
Groisman and Lesgus, who commented on the study, said this points to learning-related plasticity being governed by these really local interactions on the apical dendrites.
Got it. So apical is local. What about the basal dendrites?
Basal dendrites are more like the bushier branches closer to the base of our tree near the cell body. They tend to gather input from wider areas, maybe deeper brain regions.
Bushier, wider input.
Exactly. And the synapses on these basal dendrites behave differently. Their strengthening or weakening seem to be more in step with the overall activity of the whole neuron, a more global rule, you could say.
So less about the immediate neighbors, more about how the whole neuron is doing.
That seems to be the pattern they observed. They even did an experiment where they blocked signals from the main cell body. That affected the basal dendrites activity, but interestingly, not the apical ones.
Really? So the apical ones were acting independently.
It suggests the significant degree of independent function. Yeah.
Okay, that's a striking division of labor. Basal looking at the big picture, apical focused on the microlocal stuff. This sounds like it could tie into a big challenge in neuroscience, the credit assignment problem.
Absolutely. That's a key connection the researchers made. The credit assignment problem is basically asking, how does the brain know which specific synapse change was actually useful?
Right, if a neuron fires and something good happens, how does it know which of its thousands of inputs contributed to that success? Especially when synapses mostly just see local information.
Exactly. It's like you succeeded at a task, but how do you give credit to the specific tiny adjustments that led to success? It's a huge puzzle.
And this study offers a clue.
It suggests a potential solution. Maybe the brain doesn't solve it with one single mechanism. Maybe different compartments handle it differently,
So the apical dendrites assign credit locally.
based on those tight local interactions, and the basal dendrites assign credit more globally, based on the overall neuron's success signal.
That makes a lot of sense.
Yeah, the senior author, Takaki Komiyama, summed it up by saying this discovery fundamentally changes how we think the brain solves credit assignment. He described it as individual neurons performing distinct computations in parallel in different subcellular compartments.
So one neuron is like a team of specialists working together, but with different jobs.
Kind of, yeah. Not just one single processor anymore.
This is, well, it's huge, especially thinking about memory. For anyone studying intensely, like for the UPSC, memory is everything.
It really is.
Yeah.
And this gives us a much more granular picture of how memories might actually be encoded at the level of a single neuron. It's not just a memory being stored, but potentially different aspects or associations being processed and strengthened according to these different rules in different parts of the cell.
It makes you appreciate the complexity. Groisman and Letskis had that quote about memory acquisition and retrieval being essential and fascinating.
Yes, and this research adds another layer to that fascination. For someone deep in UPSC prep, it underscores that memory formation isn't monolithic. Maybe different study techniques engage these different dendritic learning rules in different ways. Speculation, but interesting.
And what about sleep? We hear about memory consolidation during sleep, that offline learning. Does this fit in?
It potentially could, yeah. The idea that these apical dendrites, with their local learning rules, might function as somewhat independent memory units. Well, that could be really important for how fleeting experiences get solidified into lasting memories, perhaps during sleep, when the brain replays patterns.
So getting enough sleep is maybe even more critical for locking in that complex UPSC material.
The neuroscience definitely points towards sleep being absolutely crucial for consolidating learning, and maybe mechanisms like these are part of why.
Let's shift gears slightly. Artificial intelligence. AI neurons are usually much simpler, right? Just one unit, one set of rules.
Typically, yes. Most artificial neural networks use simplified point neurons that sum up inputs and apply a single activation function. They don't usually have this kind of internal compartmentalized structure with different learning rules.
So could findings like this inspire better AI, more brain-like AI?
That's definitely a hope. If we can build artificial neurons that mimic this compartmentalized processing, this ability to handle information differently in different dendritic branches, it might lead to AI that's more powerful, more flexible, maybe learns more efficiently. It's an active area of research.
Back to that neural forest analogy, the researchers used diverse branches, different hubs. It really captures this internal complexity.
It's a great visual, isn't it? A single cell acting like a mini ecosystem.
But even with these discoveries, there are still unanswered questions, I imagine.
Oh, absolutely. Loads. Like, what kinds of information are these different dendrites typically receiving? Do apical dendrites get more top-down signals and basal dendrites more bottom-up? We don't fully know the specifics of the inputs.
And you mentioned some rogue synapses on the apical dendrites, the ones that ignored the cell body signal.
Yeah, what's going on there? Why are they seemingly independent even from the main cell signal in that experiment? That's a puzzle.
Also, this idea of branches as independent memory units, does that mean a single neuron can store much more information than we thought?
That's a fascinating implication, right? If each branch or segment of a branch can store information somewhat independently, it could dramatically increase the brain's storage capacity. Still a theory, but a powerful one.
And finally, the potential link to brain disorders.
Yes, that's crucial. Understanding these fundamental rules of synaptic modification is key. If we know how it works normally, We can then investigate how these rules might go wrong, how they might be disrupted in conditions like Alzheimer's or autism, maybe addiction, PTSD.
So figuring out the basic science could lead to new treatments down the line.
That's the long-term goal. As William Wright said, understanding the basics could help us figure out what goes wrong in these different diseases.
Okay, so let's try and pull this all together. This deep dive, drawing on work highlighted by Singularity Hub from that science study, it shows us a single neuron is way more complex, more dynamic than the old picture.
Much more. Not just a simple switch.
It's got internal compartments, especially these dendrites, operating with different learning rules. Apical dendrites doing local syncing, basal dendrites tuning to the global neuron activity.
Right. Distinct computations happening in parallel within one cell.
And this has potentially huge implications for how we understand memory and coding, which is so critical for learning.
Especially for intense learning like UPSC prep.
For how the brain might solve that tricky credit assignment problem. And maybe even for designing future AI.
It touches on a lot of big areas.
So for you, our listener, especially if you're navigating the complexities of the UPSC syllabus, knowing that your brain cells themselves are performing these intricate, multi-layered learning processes.
Well, it's kind of amazing, isn't it?
It really gives you an appreciation for the machinery you're working with.
And maybe understanding these fundamentals can subtly inform how you approach studying, how you think about consolidating information.
Definitely food for thought, which leads to maybe a final point to ponder.
Go for it.
If different parts of just one neuron learn in different ways, local versus global, maybe processing different kinds of inputs, what does that suggest about how we learn as whole individuals? How do we integrate all the different kinds of information, the different experiences, the abstract concepts, and the concrete details?
How does our whole brain, built of these complex units, weave together such different threads of learning? Exactly. It's a big question, but thinking about the complexity at the single neuron level really makes you wonder about the larger scale.
That's a great thought to end on. If you found this deep dive into the brain's learning secrets fascinating, please do like, subscribe, maybe leave a comment. Let us know if you have questions or ideas for other topics.
We'd love to hear from you.
Thanks for joining us on the deep dive.